Click for IB Biology. Here we're looking at post-translational modification of proteins, modifications of polypeptides after their initial synthesis. We'll be looking as our example at the conversion of pre-proinsulin into the active insulin. So pre-proinsulin is the first protein which is produced, and then that is activated into the form of insulin. The production of insulin takes place in the beta cells of the pancreas. The gene, which is in the nucleus, is on chromosome 11. It's actually just on the very end of that chromosome in this position here, and produces a pre-messenger RNA molecule with 2 introns. And in order to create the mature messenger RNA for translation, these 2 introns are removed. Once translation has occurred, this produces a polypeptide of 110 amino acids. This polypeptide is known as Preproinsulin, and this is produced by the nucleus, the transcription, and then the translation of the gene. So now we focus on the protein itself. So if we look at those 110 amino acids, the first section is known as the signal sequence, which is composed of 24 amino acids, and this is at the N-terminal of the polypeptide. Then there's the so-called beta chain, which is composed of 30 amino acids, a 31 amino acid C-peptide, and ending in a 21 amino acid alpha chain. Now, the beta and the alpha chains are carrying amino acids that will eventually allow them to form disulfide bridges in their structure, but at this stage that has not occurred. Now, the first step in the modification of this protein here, this is pre-proinsulin that is produced on the ribosome of the rough endoplasmic reticulum, because we remember that we know that the insulin will be secreted outside of the cell, so it must be the rough endoplasmic reticulum. And so the signal allows the protein to enter the rough endoplasmic reticulum, so this signal structure here. And as that occurs, there's a protein, an enzyme in the membrane, which cuts off the signal, and this is called signal cleavage. And so our pre-proinsulin has the signal removed, so that in the rough endoplasmic reticulum, we now have proinsulin. So the first step is the movement from the cytoplasm into the endoplasmic reticulum, the breaking off of the signal 24 amino acids, and we're now at proinsulin levels. The proinsulin is now in the rough endoplasmic reticulum, and this is then going to be moved to the Golgi apparatus. And in the Golgi apparatus, what will happen is that the C-peptide is going to be removed. That's the first thing. And then also inside the Golgi apparatus, I've just drawn this outside so you can see it, is that the disulfide bridges form between the beta and the alpha chain on this side. Now, this part here is 30 amino acids. This part here is 21 amino acids, meaning that our insulin is actually 51 amino acids. So when we count it all up, we find that we're actually 2 or 3 amino acids short, and this is because when the bonds are broken here, amino acids are actually lost to the system. So here's an image here of the insulin molecule from the protein database. So we've gone from proinsulin in the rough endoplasmic reticulum to insulin in the Golgi apparatus. So this is our modification. And then we step back to look at what's going on here, so that the Golgi apparatus then will have the insulin packaged in secretory vesicles, which will come to the cell membrane, merge with the membrane, and deposit the insulin into the plasma. And this is the secretion of insulin from the beta cells of the pancreas associated with the regulation of blood sugars. Note also that at the same time that the insulin is dumped into the plasma, the C-peptide is also released. Now, there is no known function of C-peptide in the circulatory system, but C-peptide is used as a marker for insulin levels. So our original protein has been modified in two steps, one in the endoplasmic reticulum and the second one in the Golgi before being secreted in its functional insulin form. As we go through proteins, lots of proteins are in the circulatory system. The next video is linking to how proteins are recycled when their function is finished. There's a link to the channel and there's a link to the playlist.